my name is Casey and I will be talking about the enzyme lab that I worked with my group members. The other people in my group was Carly, Tatiana, and Melinda. So first we're going to go to the introduction of our enzyme that we tested during this experiment. The enzyme that we are testing is the enzyme trypsin. Enzymes, first of all, lower the activation energy of reactions to catalyze a specific chemical reaction. Now trypsin is a protease. It is found in the small intestines of humans and is one of the several proteases that digest dietary protein. And the conditions that it is found in the small intestines is a pH of 8. The average temperature that trypsin is found to work at is between 37 degrees Celsius and 50 degrees Celsius. And the salt concentration for that is highly variable. So next we're going to go into our introduction to our experiment. The condition that we are testing is pH, specifically how the pH level from 4 to 10 affects the trypsin digestion of the substrate azocasein. Our experimental question was, how does that basic pH range from 4 to 10 affect the trypsin activity? Our hypothesis was that trypsin activity will be optimum at a more neutral pH range of 7, 8, and 9, and decrease at a lower pH of 4, 5, and 6, as well as a higher pH of 10. A rationale behind this was that since trypsin is found in the small intestines at the pH of 8, it works very well in this condition. So if it varies from that, either decreasing or increasing that pH, it will decrease in the activity. So next we're going to go into our protocol of this experiment. Our experimental variable is pH. Our experimental conditions is a pH of 4, 5, 6, 7, 8, 9, and 10. Our control variables was temperature at 45 degrees Celsius, which was between that 37 degrees Celsius and 50 degrees Celsius that trypsin is found to be the most active in. And then the salinity that we had was 0.15 molars. Our negative controls, we had 3 of those, which was pH of 4, 7, and 10, which gave you an acidic, basic, and neutral range. And in these negative controls, we did not add the trypsin. We wanted to see if the substrate azocasein would go through any digestion without the enzyme assisting it. So we also did 3 replicates of the pHs, all 4 through 10. We wanted to make sure that there weren't any outliers and get a good average. So next we're going to go into our experimental steps. For each experimental treatment, we added 450 microliters of the reaction buffer, and this was to establish the pH. We added 500 microliters of azocasein. After that, we placed it in a water bath for 5 minutes to bring it to that 45 degrees Celsius. We took it out and added the 50 microliters of trypsin. Unless it was the negative control, we did not add the trypsin in. We then placed it back into the water bath for 20 minutes. We brought it back out, added 500 microliters of 10% TCA, and the addition of the TCA was to stop the trypsin digestion of azocasein. We then centrifuged the test tube for 2 minutes to break apart the digested azocasein and the supernatant. We then take the supernatant and transfer it to a clean cuvette, which we then put into the spectrophotometer, and then read the absorbance for each to 440 nanometers. So next we're going to go into the data that we got from our spectrophotometer. So you can see here we've got the pH of 4 range through 10. We've got the 3 replicates for each of the pH ranges, which our average was here below. If you notice closely, it slowly starts to increase until we reach a max of 0.557, which was our highest at a pH of 9, and then it started to decrease again at 10. Our negative controls were listed down below with their averages there, and those were all pretty similar in their averages. And below we have our t-test to see if there was any significant differences between them. We tried to see if there was any significant difference between the pH of 9, since that seemed to be the highest absorbance level. there was significant difference between pHs of 4, 5, 6, 7, and 10, which was less than 0.01, and there was not a significant difference between the pHs of 8 and 9. So here below you can kind of see it a little bit better on the graphs. You can see how it starts to increase from 4 all the way up to 9, and then decrease down at pH of 10. And then over here we have the negative controls, which are all have very similar absorbance level. All right, so then we're going to go into our results. Now this was more of a visual result. So you can see how pH of 4, it was fairly clear. It had a very big pellet, which was collected down at the bottom, and this was the undigested azocasein. As the trypsin activity increased at a pH of 9, it was a smaller pellet, and it became a darker yellow. On the negative controls, it was a light yellow or a clear, and had a very large pellet down at the bottom. So next we're going to go into our conclusion. So our results were consistent with our hypothesis that the trypsin activity will be most effective at a more neutral pH. In our hypothesis, we stated as the pH becomes more acidic from 4 to 6 and more basic at 10, the trypsin activity will decrease. The peak digestion for azocasein would be at pH from 7 to 9. And we did confirm that it was better at 7 to 9, specifically at 8 and 9. The results from other groups, they also tested pH as well. The other groups tested the pH, found that the more neutral ranges, 7, 8, 9, were also where trypsin was most effective. So some potential improvements that we have was to increase the range of the pH even higher than 10 to see if the trypsin continued to decrease or if it went back up again. So you could see how at 9 it was increased the most and it started to decrease. We wanted to see if that would continue the trend. Application to bacterial growth. Since trypsin is found in that neutral pH of 8, 9 in the small intestine, it is found in our experiment that it is the most effective at 8 and 9, which is our conclusion. So, and then down below we have our references here. And thank you for viewing.